All right, this lesson is going to be about cell growth and division, and specifically about this process we call mitosis. These notes go on page 25 in your notebook. Now, we've been talking a bit about cells, and you need to know that cells, when we look at all those organelles, they're not just sitting in one place. Cells are very active places. Lots going on in there. All eukaryotic cells, remember cells that have a nucleus and all these cellular organelles, they have a life cycle with different stages in it. And we call the whole cycle of a cell's life the cell cycle. Kind of makes sense. We're going to talk about the 4 stages of the cell cycle now. They are G1, S, G2, and then finally mitosis. So starting with G1, G1 stands for gap 1. This is the gap cycle, or the gap stage, I should say. In this stage, the cell is carrying out its normal functions. So if it's a skin cell, it's just doing what skin does. If it's a brain cell, it's doing what brain cells do. It's doing its job as the cell that it is. Most of a cell's life, it's going to be in gap 1, in G1 stage. It's going to spend most of its time in this stage. Some cells, most cells, at some point will leave G1, leave gap 1, and enter the S phase. S stands for synthesis. Synthesis means to make something. So in this process, in this stage of a cell's life, it will first receive a signal to enter this stage, and then during the time that it's in this stage, what it's doing is it's copying the DNA. Remember, the DNA is the instructions for how to make everything that the cell's doing and making. So during this time, the cell's going to make a whole copy of its DNA. Then the cell will enter G2 once it's done copying the DNA. G2 is called gap 2. That's what it stands for. During this time, there's additional growth of the cell. And again, there's another critical checkpoint before the cell will enter mitosis. So at the end of gap 1, there's a signal that says, okay, make the copy of the DNA. And at the end of G2, there's a signal that says, time to begin mitosis. The final stage that we're going to talk about is mitosis. It's sometimes called M. Just for mitosis. At this time, the cell separates its 2 sets of chromosomes, or its 2 sets of DNA, and it will form 2 new cell nuclei. At the end of mitosis, there's a process called cytokinesis, which we will talk about in a minute, and that's when the cell actually splits in half. The 2 new cells will go back and enter G1 again and start all over again. So even though this looks like a process going in one direction, it's actually a cycle. So here's an overview of what I just talked about. The beginning of a cell cycle starts with G1. The cell will spend most of its time in G1. At some point, it will receive a signal to enter S phase. At the end of S phase, it has duplicated its DNA. It will enter G2, gap 2. At the end of G2, it will receive a signal to begin mitosis. And this isn't really even that accurate, because mitosis, usually the cell spends very little time in mitosis. Let's talk for a minute about why cells are dividing anyway. In some unicellular eukaryotes, such as Protists, amoeba, or something like that, cell division is how they reproduce. So they might divide, make a new cell, and now they've reproduced. In multicellular organisms, such as yourself, cell division helps them to grow. Okay, so as you grow larger, you grow more cells. You don't grow bigger cells. And we already talked about why cells have to be small. Another thing they could do is repair tissue. So if you get injuries of any kind, an injury or a cut or something, it's mitosis that allows you to repair that cut and replace those cells. And in some organisms, they can replace whole tissues and organs this way. So here's an example of that. This is a newt. A newt. A newt is not like a human. If they lose an entire limb, they can regrow it. So here's after a week. They've lost a limb. Here's after a week, they've grown a little nub. After 3 to 6 weeks, it's looking more like an arm. And then after 6 to 9 weeks, they can regrow the entire limb. And this, of course, is due to this process of mitosis. Now we're going to focus in a little bit on mitosis. And we talked about G1, S, and G2. Together, these are called interphase. So this whole time period that the cell is not dividing, not actively dividing, we call that interphase. Mitosis itself is divided into separate stages, which we will talk about in just a minute. So let's talk about interphase. During this time, G1, S, and G2, if you're looking at cells in interphase, you can clearly see the nucleus present. These are like the cells we've looked at in class. The nucleus is clearly present. You can see the nucleolus as a dark spot here in this diagram. And you can see the chromosomes are this thread-like chromatin. This is the DNA. It's just kind of spread out like spaghetti, like pasta, okay? And you can just see it kind of spread out. You don't necessarily see chromosomes. At the beginning of mitosis, the first step is called prophase. During prophase, this spaghetti-like chromatin, the DNA, is going to condense into these chromosomes, and they become visible. Each chromosome is really a pair of sister chromatids. They're paired up because they've been copied during the S phase. The nucleus disappears, and then these structures called centrioles, which I think you've seen in diagrams, but I never talked about them, these centrioles begin to move to opposite sides of the cell. So here we have prophase. During prophase, you can see the nucleus, the nuclear membrane is beginning to disappear, and you begin to see these dark chromosomes showing up. And again, here's a plant cell over here. You can see the dark chromosomes showing up. So the chromosomes appear, and the nucleus starts to disappear. Here are the centrioles, and they're beginning to move to opposite sides of the cell. The next stage is called metaphase. During metaphase, the centrioles send out these things called spindle fibers, these little fibers that are going to attach to the chromatids or the chromosomes. They attach to them, and they begin to pull on them and line them up along the center of the cell. Now, the purpose of this is to get all the chromosomes lined up because they're going to be divided out. Just like you're picking teams when you're playing sports, you got to pick teams like in PE class or something. You want to line up everybody so that you can make sure everyone's accounted for. So here's what metaphase looks like. In metaphase, you can see the chromosomes are beginning to line up along the center of the cell. Here's an animal cell, and here's a plant cell. You can see the chromosomes lining up. Thick coiled chromosomes are lined up in the center of the cell, and spindle fibers are attached. So here are the spindle fibers. Here's the centrioles on either side, and then you've got your chromosomes lined up in the center. The next phase is called anaphase. During anaphase, the sister chromatids are going to be pulled apart by the spindle fibers. So they've lined up during metaphase, and now they're going to be pulled apart, one to each side. Each sister chromatid moves to the opposite sides of the cell, so that each new cell is going to get exactly the right number of chromosomes. Here's what this looks like. Now you've got your chromosomes being pulled apart here in the animal cell, and here in the plant cell, the chromosomes are being pulled apart. Chromosomes have separated and are moving toward the poles. Here's your centrioles pulling each sister chromatid apart so that each new cell gets exactly the right number of chromosomes. During telophase, the next phase, this is actually the final phase of mitosis. Each newly forming cell now has a complete set of chromosomes, and a new nucleus begins to form around each new cell, so the process is ending. Here we have telophase. The chromosomes are now at the poles. Each centriole is at either side now, and this is going to become two new cells. Here we have an animal cell doing it. You have the chromosomes pulled apart, and now a new cell forming on either side, and here are plant cells doing the same thing. And finally, when mitosis is complete, there's a process called cytokinesis. Cyto means cell, and kinesis means to move, so this is cells moving apart. This is the rest of the cell contents being divided into the two new cells. So far we've just talked about the nucleus being divided during mitosis. Now we have all the rest of the cell components being divided up. All the mitochondria, all the ER, everything has to be divided up. In animal cells, there's a trench that forms, and then it pinches off to form two new cells. In plant cells, a new cell wall has to be built between the two new cells. But either way, you get two new cells forming. So this is what this looks like. Again, this is a picture we've seen before, but there's a trench forming between these two animal cells, and they're going to pinch off. Here we have plant cells, and you have cell wall material being added between these two newly forming cells until finally, this is called a cell plate, finally a whole new cell wall is created between them, and again, because of mitosis, you have a new nucleus in each new cell. So this is a way that cells can make new cells and make sure that everything the new cell has is there, that belongs there. All the cell organelles, and especially all the chromosomes. All right, we'll see you next time.